Dear students, our lecture today is the gene theory. As you know, the world today is concentrated on the use of genes in the medicine. We can use gene in the treatment of disease and instead of drugs. What genes can do? Genes which are carried on chromosomes are the basic physical and functional units of heredity. As you know, genes are coding for all biological activities in our life. The movement, the motility, all of these are controlled by genes. Genes are a specific sequence of bases that encode instructions of how to make a protein. Is the protein that perform most life function and even make up the majority of cellular function. Now, why genetic disorder? When genes are altered so that the encoded protein are unable to carry out their normal functions, genetic disorder can result. As you observe here, the X-linked recessive genetic defect from daughter from father to the offspring. Here, the defective genes was transferred from father to the offspring, and the XD. D means defective gene. Defective genes will be inherited here from the father to the offspring, while the defective gene XT will be inherited here from the mother to the offspring. Now, we must know that all of us here carry some defective genes. Some of them are apparent and the other are inapparent. Each of us carries about half of a dozen defective genes. We remain blissfully unaware of this fact unless we or one of our close relatives are amongst the many millions who suffer from genetic disease. About 1 in 10 people has or will develop at some later stage an inherited genetic disorder, and approximately about 2,800 specific conditions are known to be caused by defective mutation in just one of the patient's gene. Now, all of you now in this class inherited the genetic information from father and mother. Half of the genetic information are inherited from dad, and the other half will be inherited from the mom. Now, most of us do not suffer any harmful effect from our defective genes because we carry 2 copies of nearly all genes, one derived from our mother and one we inherited from our father. The only exceptions to this rule are the genes found on the male X or sex chromosomes. Males have 1 X and 1 Y chromosomes, the former from the mother and the latter from the father. So each cell has only 1 copy of these genes on these chromosomes. The law of inheritance. In the majority of cases, 1 normal gene is sufficient to avoid all the symptoms of disease. Just 1 normal gene would be enough to avoid all symptoms of disease. If the potentially harmful gene is recessive, then its normal counterpart will carry out all the tasks assigned to both. Only if you inherited from our parents 2 copies of the same recessive gene will a disease develop. Now, after we take all the introductions, all the necessary informations in this simple introduction, now we must understand what does it mean by gene therapy. Gene therapy is the insertion of genes into an individual cells and tissues to treat diseases such as a hereditary disease in which all deleterious mutants allele is replaced with a function One, although the technology is still in its infancy, it has been used with some success. What are the goals of gene therapy? First, a normal gene may be inserted into a non-specific location within the genome to replace a non-functional gene. This approach is most common. An abnormal gene could be swapped for a normal gene through homologous recombination. And the abnormal gene could be repaired to selective reverse mutation, which returns the gene to its normal state, to its normal function. And the fourth goal of gene therapy, the regulation, the degree of which a gene is turned on or off of a particular gene could be altered. Yes. Now, what are the uses of gene therapy in medicine? We must understand when and where we can use genes in medicine. First, replace missing or defective gene. When I have a defective or missing gene in a specific location, we can use gene therapy to replace missing or defective gene. Second, to deliver genes that speed the destruction of cancer or tumor cancerous cells. Also, to supply genes that cause cancer cells to revert back to the normal cells. Thus, when we ask some patients when the stage of cancer, where the stage of cancer is happening, that's in the normal cells. The tumor genesis theory started from conversion of normal cells to transform stage, and from transform stage to the malignant stage. If the patient in the first stage, from normal to transform stage, this stage is reversible. That means what? That means the patient can repair to the original state. But if the patient from the stage from transform to tumor to malignant cells, in this stage it's irreversible. That means what? That means the malignancy will not be reversed to the original stage. The patient cannot revert to the original state, and it's so difficult to treat. Okay? Thus, deliver bacterial or viral genes as format of infection, as a form of vaccination. Deliver bacterial or viral genes as a form of vaccination. And to provide genes that promote or impede the growth of new tissue, and to deliver genes that stimulate the healing of damaged tissue. Yes. How it works? A factor delivers the therapeutic gene into a patient target cells, and the target cells become infected with the viral factor. After that, the factor genetic material is inserted into the target cells, and the functional protein are created from therapeutic gene, causing the cell to return to a normal state. Yes. Gene therapy is experimental. Advances in understanding and manipulating genes have set the stage for scientists to enter a person's genetic material to fight or prevent disease. Gene therapy is an experimental treatment that involves introducing genetic material, DNA or RNA, into a person's cells to fight disease. The majority are right here. As you show in this figure, gene therapy of cancer. Gene therapy is being studied in clinical trials, research studies with people for many different types of cancer and for other diseases. It is not currently available outside clinical trials. Thus, in this case, the specified retrovirus and the introduction of defective gene into the gene here, retrovirus carry normal human gene, and the cancer cell infected with retrovirus. As you show here, the retrovirus attack cancer cells and lymphocyte enter cancer cells to be called cancer cells. Here, the cancer cells will be break down. Why? That's due to what? That's due to gene therapy. Gene